Ooh, I'm the host. Hello, people. Nice to see you all again. Those of you with your Amazon are looking chipper. I'm Robin Russell. I'm only catching your eyes. I'm Robin Russell. Teasing you, teasing you. What do we got so far? 35. Let's wait a couple minutes. Sydney Barrier. This is your 500th class with me now. You're getting a minor in Slish too, whether you like it or not. Trevor DiCarlo. I've not seen you in a while.
I feel like I've been in one of your classes for the last like 3 semesters.
Yeah, you've been lucky up to now. It's been good. DeJovany Jupiter showing up again. Good for you. Glad to see you there, DeJovany. Love your name, by the way. Next time you see your parents, tell them that your cell biology professor likes your name. I'm sure it'll matter to them.
Sure, I will.
They're really going to give a shit, I know. 11:02. 11:02. Rock and roll. Let's do it. Let's do it. The squares stayed home, right? Okay. All right. So today we discuss skeletal muscle and cardiac muscle, structure and function. So I always like to show this, just a thing I do. If you ever take a rope and take it apart, it's like it's not one solid fiber. It's always like a whole bunch of fibers that are made into a little string. These strings are wound up together to make cables. These cables are put together to make ropes, and the ropes are tied together to make a really big, thick thing. So that structure makes it a lot stronger. It's harder to rip that apart than if it was a single cable. And that's why even with like electricity, if you look at wires, they're usually not one thick piece of copper. They're a whole bunch of little copper threads together, and that gives it more strength. So same thing going on here with your skeletal muscle. The whole muscle starts from origin to insertion, and each muscle cell goes the whole length of the muscle. And where is it? This is a muscle cell, although normally it has a plasma membrane on top of it. They've stripped that off. These are nuclei. Skeletal muscle cells are multinucleated. And the reason for that, I believe anyway, is they start off as shorter cells that then fuse together and become longer cells, and they keep all their nuclei. So it's a multinucleated cell. So you've got plasma membrane over this, and inside the cell itself, it are the myofibrils. Oh, I see. Each of these is a muscle cell. That's right. Let me back up again. You got the whole muscle, then you've got individual muscle cells. Inside the muscle cell, you've got myofibrils. So you've got kind of that cable string thread kind of structure there. So the myofibril then is the contractile part of the skeletal muscle, and each myofibril is composed of kind of an array of sarcomeres. And the sarcomere is the contractional unit of the skeletal muscle. When I say contraction, the contractional unit, I mean nothing smaller than this can contract. It needs all of this structure here to be contractible. Contractible, there's a new word. I love making new words. Okay, so here is our sarcomere. Sarcomere goes from Z line to Z line. And if you cut this in a cross section and look at it, here's your thick filament, and you've got 6 thin filaments around it. So let's back up a couple. Remember your thick filament here, these myosin heads kind of spiral around in three dimensions around the thick filament. And so you always have a myosin head that's facing a thin filament somewhere. So typically when we talk about the sarcomere, it looks two-dimensional, but in reality, it's three-dimensional, and you've got myosin heads interacting with thin filaments all over. So each thin filament interacts with two thick filaments. Each thick filament has got 6 thin filaments together, and that's your structure. And so if you're someone who plans to take a standardized test in the near future, and it involves biology, they always ask you this stupid question about skeletal muscle bands. Which bands get shorter during contraction? Which ones do not? And I don't know if they still ask that question, but they always used to. It drives me nuts because it's pretty lame question in the end, because it's based on when people were looking at skeletal muscle through the light microscope and they didn't know anything about the proteins there. All they could see is they were striated or striped. And you could see some of these stripes got smaller during contraction and some didn't. Some stayed the same length. And that was kind of like a clue as to how things work. We know everything about skeletal muscle now. So I don't know why people still focus on the which bands are which bands, but anyway, they still do. So let's talk about it real quick. So your bands then are the H zone in the middle. So you've got the M line directly in the middle, and the H zone is where you've got myosin that's not overlapped with actin. So as the muscle contracts, that H zone gets bigger. I mean, as it relaxes, the H zone gets bigger. As it contracts, the H zone gets smaller because you increase the overlap during contraction. Then you've got the A band, which is the whole length of the myosin thick filament, and that's not going to change during contraction. The length of the myosin filament doesn't change during contraction. Then you've got the I band, which is thin filaments that are not overlapped with myosin. And so that's going to shorten during contraction and lengthen during relaxation, because as you get overlap, you've got less unoverlapped. That's a great word too. I think I made that one up today. Unoverlapped thin filament. And then at the end of the thin filaments, you've got the Z line. So you've kind of got your plus end of your microfilament here and your minus end here. And this is kind of like a head-to-head arrangement with the next sarcomere, because this is the plus end of this microfilament and the minus end would be over here. Okay, so H zone in the middle, M band in the middle there, A, I, and the Z line. And this overlap here is where you get the cross bridge cycling. So the cross bridge is the connection between the thick filament and the thin filament. And that cross bridge is actually the head of the myosin reaching over and grabbing the actin. All right, so that's the business side. That's what causes the contraction. Another thing that's important is all the other structural proteins involved that hold this in place. You can imagine if you've got a thick filament and thin filament that are supposed to slide past each other, and the thin filament does this, well, now you're not contracting anymore, right? Or if the thick filament moves and it's no longer, efficiently grabbing a thin filament, it stops contracting. So all these other proteins here are involved in holding those thick and thin filaments so they can slide past each other perfectly. So starting in the middle, the M line, I think it's called the M line because you've got myomasin there, structural protein around the center of the thick filament. Oops, damn it. Damn your eyes. Okay, myomasin there. And then you've got this titin that goes across the whole length here of the this half of the sarcomere and attaches to the Z line. And so that titin then is kind of holding this thick filament in place, making sure the myosin heads are lined up properly, that they can grab the thin filament. So that's titin. At the plus end of the thin filament, you've got CAP-Z. If you remember from chapter 13, CAP-Z captures the plus end and stops it from depolymerizing. It may also be involved in holding that thin filament onto the Z line. The Z line's got to be proteins too. They're not mentioned here in your textbook, but obviously everything is a protein. The other end of the thin filament then is held by tropomodulin, so you're not going to depolymerize from this side. So the thin filament is stabilized at either end by tropomodulin and CAP-Z. And then you've got Mr. Nebulin here that is, again, a coiled coil. Remember that structure. a coiled coil that wraps around the thin filament, again, keeping it straight, and making sure that it stays perfectly aligned to interact with your thick filament. So that's your sarcomere structure. Okay, so how do we control this interaction? So the control of contraction is by controlling interaction of the myosin with the actin. And we're doing that by controlling the actin. So here, this blue is the actin, and the green is tropomyosin, a coiled coil. Have you heard of that before? So this is another coiled coil, and it is covering the myosin binding sites. So right underneath this is where myosin would bind in order to cause contraction. So if this is in this position, then myosin can't interact and the muscle relaxes. And the position of this tropomyosin is controlled by the troponin complex. Troponin T, troponin I, troponin C. So the troponin complex, when there's calcium around, calcium will bind to the troponin C subunit, and this will change conformation. When it changes conformation, it also changes conformation of the tropomyosin, pulls that tropomyosin off the myosin binding sites, and then myosin and actin can interact. And as long as you've got ATP, myosin and actin together, myosin will grab actin and cycle and cause contraction. So really, with the skeletal muscle, if you strip the plasma membrane off, all you have to do is hit it with calcium and it would contract. And ATP, because you take the plasma membrane out and the ATP will diffuse away. So if you had, they used to call that a skinned muscle prep, where you take the muscle and you get rid of all the plasma membrane and then add back to it different things and see what causes the muscle to contract. Turns out you need actin and calcium at that point. Okay. All right, so cross bridge cycling. We're going through this kind of quick. I will talk slower so class isn't done early. Let's start here. This is called the Riger complex. The myosin head is bound to the actin and it won't let go, basically. It's bound really tightly there, will not let go. And that's the Riger complex. Oh, damn it. All right, when there's ATP around, and the head of this myosin right now is at a 45-degree angle with respect to something. I don't know, I read the 45-degree angle somewhere, but I'm not sure what that's with respect to. But anyway, keep in mind, this is at 45 degrees. I guess this is 45? No, it can't be. But anyway, ATP binds and the myosin head lets go. Okay, now you're in the normal state of a relaxed muscle where it can be stretched or contracted, stretched or pushed together, unstretched, flaccidly, right? ATP is attached to it. There's no connection between the thick and thin filaments. All right, but the next step that happens is this myosin head will phosphorylate itself with the ATP. So boom, it sticks phosphate on itself. And you know that third phosphate in ATP is high energy. So as it phosphorylates itself, it kind of absorbs the energy from that phosphate. And the energy from the phosphate then causes the head to move to this 90-degree position. Again, I don't like this figure because they're not showing the percentages or the degrees the way they should. But anyway, this is your 90-degree conformation. Now, this conformation is your high energy conformation because the energy from that third phosphated ATP was absorbed into the myosin muscle and it went to a high energy conformation because now it's at higher energy, okay? So now it's like a coiled spring there. The next thing it does is let go of the phosphate, and when it releases the phosphate, it grabs onto the actin, okay? And now the final step is when the ADP comes off, then it goes back to its low energy conformation. And in going to that low energy conformation, it is moving that thin filament forward. So that's what we call the power stroke. So this step from here to here is the power stroke. The ADP comes off and the myosin goes back to its low energy conformation. So just to go around the circle again, myosin attached, Riger complex, ATP comes, it lets go. It phosphorylates itself and it goes into the high energy conformation. Phosphate comes off, it grabs the actin, and then ADP comes off and it goes back to its low energy conformation and moves the thin filament in the process. Okay, questions, comments?
So, I'm sorry, I'm a little confused about this, the myosin head. Does it attach to the sarcomere, or does it like barely even touch it?
It directly binds to the thin filament, to the actin. Okay, the actin has got like a spot on it where the myosin can grab, and that's all about, you know, what amino acids are on the actin, what amino acids are on the myosin head. They perfectly fit, so they grab and they bind tightly. Okay, and then when it moves, it grabs, it pulls it with it. Okay, does that make sense?
Yes.
Okay. And in fact, they grab so tightly that right here, if you're dead and you got no ATP, that's where your muscles end up. Okay. When you die in your muscles, you've got less ATP in your muscles, your calcium stores will run down, which means calcium is going to come into the cell and the cellular calcium will increase. All your troponin will be moved. And so myosin can interact with actin. But then, and so it does, but then the actin runs out, or the ATP runs out, and your myosin is stuck to that thin filament, and that's rigor mortis, right? If someone's dead for, I don't know, I think it takes an hour or two, but once they're dead for a little while, their muscles get really stiff, and that's not an active contraction. That's a passive connection here between your myosin and your thin filament. Can't let go because there's no ATP.
So are you saying once you die, the muscles contract for a short amount of time until they just stop?
Well, I wouldn't say you're going to die. I think you're going to live forever. Just keep that in mind. But when I die, I don't know if they're actually going to contract. I don't think so. No, they won't contract. But basically, your calcium stores calcium comes out of the SR, the whole cell fills with calcium, myosin attaches to the actin, but there's no ATP, so it can't do anything. It just stiffens. I think that's kind of what happens.
Oh, so it's not moving at all?
No, it's not moving at all. No. And that's why, you show up to someone who's been dead for, 4 or 5 hours, you turn them over, they're stiff as a board, and they're in exactly the same position they were as they were laying there dying and stiffening up. It doesn't really contract, it just stiffens. Okay. Any other questions, comments? Okay, so important things to remember here are the sequence of events. Okay, I really like that because it shows you how energy is really used in a molecular process. The energy from that high energy ATP, that third phosphate, gets transferred to the myosin and it goes into this high energy state. And then that high energy gets passed to the actin kind of by changing its position. That's kind of cool. The other is you should know what the power stroke is here. You should know about the Riger complex. And that's about it.
Sorry, are you saying Riger with a G in the middle or Rider with a D?
With a G, R-I-G-O-R, Rigor.
Oh, kind of like rigor mortis?
Exactly.
Okay.
Yeah. Because after you're dead, this is where all your muscles end up, in this position. And so rigor mortis. And I think it started with, I think rigor is like Latin for stiff. So rigor mortis, stiffness after death. And then they just used that name when they learned about where the myosin heads went and everything. They call it the Riger complex because that's where you are in rigor mortis.
So I have a question. So where does all the ATP go?
Where's the ATP go? So
Yeah.
the ATP comes in here And then the third phosphate of ATP gets added to the myosin head here. So now you're down to ADP. And then that phosphate comes off. So you can connect here. And then the ADP goes off and it goes back to the mitochondria and gets rephosphorylated.
Oh no, not. I was talking about like when the person is dead and going through rigor mortis. You said it remains stiff because there's no ATP to move the head. So where does all the ATP go?
Well, let's think about death. What happens in death? So I guess death is usually considered when your brain dies, but also when your heart stops beating. So when your heart stops beating, oxygen stops going to the rest of the tissues. Now, the rest of the tissues are perfectly alive. Okay, it's just that they're not getting oxygen anymore. And since they're not getting oxygen, then they start to run down because you always have ATP being made using oxygen that is maintaining your ion concentrations in the cell with the sodium potassium ATPase, and everything else in the cells is constantly being maintained by ATP. And then, so when the oxygen doesn't show up anymore, those maintenance operations keep going until there's no more ATP around, and then the whole thing just kind of stops and all the ion gradients zero out. So you've got the same amount of calcium all over the cell and outside the cell and everything. And then that's kind of cell death at that point. Does that make sense?
It does. Thank you.
Okay. Any other questions, comments? Anyone else think they're going to die? None of you people are going to die. I plan to someday, though. Looking forward to it. Okay. Not really looking forward to dying, but I remember last fall, I was having a really hard fall last year, a lot of things going on. And I heard a report about COVID and someone had it really bad. And so in order to intubate them, they put them in a medically induced coma for a month. And I was thinking, medically induced coma, that would be kind of nice. I think I would like that for about a month. Just a little relaxation time, a little me time, you know? Anyway, back to our skeletal muscle. So here are your sarcomeres. From gray line to gray line is a sarcomere. So some of the other structural components of the skeletal muscle, there's other structural components of the skeletal muscle that are important for controlling calcium in the cell. And that's what's going to control the contraction. And it kind of starts with the plasma membrane. So the plasma membrane, of course, covers the whole cell or the whole muscle fiber, but then it's also got these little extensions called T-tubules that come down into the cell and between the muscle, the myofibrils. So everywhere you've got a myofibril, you've got a T-tubule going through, and I believe they come out on the other side. So it's a tubule that goes right through the whole cell. And those T-tubules are intimately connected to the endoplasmic reticulum, the smooth ER in skeletal muscle. Now, the smooth ER in skeletal muscle is highly developed. It's different from other cells, like a liver cell or something. And so we call it a sarcoplasmic reticulum. It's very specialized for calcium handling. So this smooth ER then we call SR, or sarcoplasmic reticulum, over here. So it's kind of a series of tubes. We used to think of it as a sac, but it's really a series of tubes. And here in these tubes, especially in the middle, you've got a protein called smooth endoplasmic reticulum calcium ATPase, the SERCA. So SERCA is a energy-driven pump that's going to take calcium out of the cytoplasm and put it into the SR. So this is one of those things that falls down when you die, because as long as you've got ATP, this SERCA is going to be constantly active, and so constantly bringing calcium out of the cytoplasm and into this ER. So this SR then gets really loaded with calcium, and the cytoplasm will be like 0.5 micromolar calcium, very, very low concentration. And inside the SR, it'll be like 10 millimolar calcium. So very high store. So that's like 20,000 times higher inside the SR than outside of it. And of course, you can't put all that calcium together in one place and without it exploding, right? All those negative charges. So you've got some calcium binding proteins in there that help to neutralize some of that charge. But they don't bind it very tightly. So you've got a huge calcium store in this SR. And you can see you've got two SRs tightly coupled to one T tubule. And we call that the triad junction. Okay, so here's your triad junction between two SRs. And here is the interaction between the motor neuron and the muscle fiber. We call it the neuromuscular junction. So your axon comes down to the muscle and then has a series of axon bulbs that form structures with the plasma membrane of the skeletal muscle. Each skeletal muscle, each muscle fiber is individually wired with the neuron. Okay, so you can contract individual muscles separately. And that's how you make a stronger versus a weaker contraction. So every time a skeletal fiber, skeletal muscle cell is stimulated to contract, it contracts as strongly as it can. So the way you get a stronger contraction versus a weaker contraction is how many muscle fibers do you recruit in that muscle. So when every muscle is contracting as hard as it can, that's as strong as that muscle can contract. When you need a weaker or slower contraction, like I'm contracting my bicep right now, but very easily, right? So you only have a few of your muscle fibers contracting. So this is called the motor end plate, where the axon bulb of the motor neuron interacts with the skeletal muscle here. And it's a very developed structure, right? Usually I'll draw it like, here's your axon bulb, here's your muscle, and there's just two membranes looking at each other. But it's actually a lot more developed than that. So you've got a really kind of crenulated plasma membrane here, and it's a little bit depressed below the level of the plasma membrane of the regular cell. So this is a structure here. So your axon bulb then will release acetylcholine from vesicles in it. And we'll talk about that process a little bit in chapter 12. But we're releasing acetylcholine into this synaptic cleft. So the synapse is the joint between the muscle and the neuron. Synaptic cleft is a minute gap between the two. So this axon bulb releases acetylcholine, and it's going to bind to receptors on the other side. And those receptors are nicotinic acetylcholine receptors. They can also be activated by nicotine, which is why they're called nicotinic. So these are nicotinic receptors, and the nicotinic receptor is a sodium channel. So when acetylcholine binds to it, the channel opens and sodium comes through. So when sodium comes through, then that causes depolarization of this motor end plate. And that depolarization then hits voltage-dependent channels in the vicinity, like over here, and that starts an action potential that goes down the plasma membrane. So the neuron releases acetylcholine, action potential, and that gets traveled up down the sarcolemma to the T tubule and then down into the triad junction. So and then to get this calcium release, what happens is in this T tubule membrane, you've got a protein called the voltage-dependent calcium channel, VDCC, and this is a skeletal muscle isoform of the voltage-dependent calcium channel. It comes from its own gene. We'll see other calcium channels later that come from different genes, but this is your VDCC in skeletal muscle. And normally, when the cell is in a relaxed state, this VDCC touches a channel in the sarcoplasmic reticulum membrane. So this is the ryanodine receptor. Ryanodine receptor is a huge calcium channel. So normally this VDCC is blocking that voltage-dependent calcium, that ryanodine receptor from opening. So this channel then, when the action potential comes down the T tubule, this will sense that change in voltage and it'll flip. So it goes from more inside the membrane to more outside the membrane. So it moves out a little bit. And when it does, it unplugs that ryanodine receptor and calcium comes flooding out. So if we back up a tad, right here is where that's happening. And this is your area of maximum overlap of the thick and thin filaments right here. So right here, you get calcium flooding out. So that calcium floods out, hits your troponin complex, pulls the tropomyosin and off the myosin actin, myosin binding sites of actin, myosin cross bridges start cycling, and the muscle contracts. And then when the action potential stops, okay, the action potential just comes and stops when it hits the end of it, okay? So it just goes boom, and you get one twitch, which is one action potential coming down, calcium channel coming out, calcium released, calcium channel going back in, calcium comes out, contraction, and then the circa pulls the calcium back in and it relaxes again. That's one muscle twitch. And that cycle takes about 1/10 of a second, I think. What's that? 1/10 of a second, 10 milliseconds. Okay, so that's how your muscle contracts. And then relaxation is basically once you stop action potentials, this goes back in, circa pulls the calcium into the SR, and the muscle relaxes. So if you want a continual contraction, you have to have continual acetylcholine release, continual action potentials, and keep moving this calcium channel and releasing calcium. So you get a stronger, harder contraction with more action potentials to that muscle fiber. Longer, I should say. Longer and therefore stronger, I guess. Okay. Any questions about this? Comments? No? Okay, so that's skeletal muscle excitation, contraction, coupling. Most important thing to remember here is what's going on right here. So the action potential comes down the T-tubule and hits that Volch-Spinner calcium channel. It's called a calcium channel, but no calcium really comes in here, or not much anyway. Nothing significant. That calcium channel is really acting as a voltage sensor. It's sensing the voltage in that T-tubule membrane. So when the action potential comes down, it moves and kind of unplugs the ryanodine receptor. When the action potential is over, it goes back into position and plugs it up again. Oh, here's another neat one. So here's it's also called the dihydropyridine receptor because it binds a drug called a class of drugs called dihydropyridine. We'll just call it the Volch-Spinner calcium channel. So there's your VDCC in the T-tubule membrane is binding with this. When it pulls back, boom, calcium comes flying out. I like to think of it as a toilet flushing. Your handle is like your VDCC. When your VDCC pulls, or that I guess it's your motor end plate now. This is I didn't look at this closely before today's lecture, but this is a quite elaborate metaphor I've built for you. This is free too. I mean, you don't have to pay extra for this kind of metaphor. Just comes with the price of the class. So anyway, here's your VDCC here, and here's your ryanodine receptor, and the tank is full of calcium. So when the motor end plate goes, the action potential goes down to that Volch-Spinner calcium channel. It moves out of place, and calcium comes out, and your poopy goes down. Okay, questions, comments about Volch-Spinner or excitation, contraction, coupling? Time is it anyway? Okay, so cardiac muscle. Cardiac muscle, in terms of the contractile apparatus, the sarcomeres and all the proteins involved in sarcomeres, basically the same. There may be some tissue-specific isoforms of some of the troponins and some of the other proteins, but structurally it's basically the same. The big difference is two big differences. One is how the action potential reaches the cell, and the other is how the action potential causes calcium release in the muscle. So starting with the action potential, I should have another figure here showing you the whole heart. So each individual muscle cell is not innervated for contraction in cardiac like it is in skeletal muscle. The cardiac action potential is inherent to the muscle itself. So one area on top of the heart called the sinoatrial node spontaneously depolarizes about once a second. Okay, maybe a little less, about once a second, and that causes an action potential to start in the atria, which pass over the atria, and then it hits this area called the AV node that slows it down, and then the AV node brings the action potential to the Purkinje fibers that then spreads up through the ventricles. Once it gets to the ventricles, it spreads cell to cell to cell. So most of the action potential spread, well, for one thing, you can take a heart out of a body, it'll keep on beating. If you keep giving it sugar and oxygen, it'll beat an awful long time. I used to do experiments with rat hearts where you take the rat heart out and quickly wash it off and then put a cannula in the aorta, and it forces fluid back down the aorta into the coronary arteries, and that fluid would have oxygen and glucose and other ions and stuff. And that would keep the rat heart beating for hours. I mean, 12 hours. You could do experiments. Usually, the heart would outlast me. I'd want to go home. So a heart spontaneously beats. You don't have to pace it at all. It's got its own pace. In fact, another funny story from grad school. I was on a list with other people in the lab to get human hearts. So when they had one guy in the lab wanted to work with human cardiac tissue. So one night we got a call at like, I don't know, midnight or so. Come on, we got a heart. So there was a drug dealer had got shot in the back of the head and decided to donate his heart to this woman who had a failing heart. So they had like two operating rooms next to each other. One was the drug dealer who was almost dead, and they took the heart out of him, walked it across the hall to the other one, and in the other side they had this woman opened up and she was on the heart-lung machine, and when the new heart came across the hall, they pulled her old heart out, put it in a bucket, brought it out to me and my buddies, and that human heart was sitting there beating. Who are you? Don't talk. Human heart sitting there beating in the pan. And I picked it up, human heart beating in my hand, got a razor blade, cut up in little pieces. So that was kind of crazy. That was one of the most fun things I did in graduate school. So anyway, that's just a way to waste time because I can see how short this lecture is getting. So cardiac. So the big thing with cardiac is your action potential is going to pass from cell to cell. And it does that by these gap junctions. So a gap junction is basically a gap junction protein on one side, the connexin, and arranges into an ion channel. And then you've got the same thing on the other side. They meet together, and now ions and small molecules can go from one cell to another. So the action potential, and that's all of these intercalated discs that connect one muscle cell to another. So the action potential then goes across this cell and goes through those gap junctions into this cell. And so we call the cardiac muscle, the ventricle or the atria, are electrical syncytia. They all work together. One action potential in one place spreads to the whole thing through these gap junctions. So that's one thing that's different with cardiac. The other thing that's different with cardiac is, hang on a sec, is your T-tubules. You do have T-tubules, but they're not as closely connected to the sarcoplasmic reticulum as they are in skeletal muscle. There's a bigger gap here. And the reason for that is the calcium channel is not directly connecting to their iodine receptors here. And it is going to act as a calcium channel. This is a different isoform. It comes from a different gene. And it is actually acting as a channel. So this is the Voltschmetter calcium channel in cardiac tissue. And what happens is when the action potential comes down the plasma membrane to the T-tubule, it opens that Voltschmetter calcium channel, and a little bit of calcium comes in. Not enough to cause contraction, but it will trigger the contraction. So we call that trigger calcium. So the VDCC allows trigger calcium to come in. That calcium comes over to the ryanodine receptor, binds to its outside domains, and that opens the ryanodine receptor. And then all the calcium that's in the SR comes flooding out, and you get a large increase of calcium in the cell. And then you get contraction, and then The circa then brings it back into the SR and you get relaxation. So that's the contraction relaxation cycle. So this mechanism we call the calcium-induced calcium release mechanism because calcium comes in and it induces calcium to release from the SR. Okay, so that's your big difference between cardiac and skeletal muscle. In cardiac muscle, skeletal muscle, which one? What am I looking at here? Okay, so this is skeletal muscle here. You've got a connection between the calcium channel and the ryanodine receptor that moves. You can see it pulling the ryanodine receptor out, boom, and opening that channel so calcium can get out. Here with cardiac, you've got a separation between the two. Calcium comes in, that activates the ryanodine receptor, and then calcium comes out. Okay. Questions? Comments? Discussion points?
I wanted to know, will we be able to tell the difference between all these cells, like the muscle cells, like in the body or in the heart or in the skeleton? How are we gonna tell those each apart?
You mean on an exam from me, or you mean in general? In general.
In general.
So in general, you can always tell a skeletal or cardiac muscle because it's striated, it's striped. Here is cardiac muscle. You can see the stripes, and those stripes are the sarcomeres. Skeletal muscle, same thing. Smooth muscle, you won't see striation, so you won't be able to tell the difference between a smooth muscle cell and some other cells. But that's the big tell-all. And your cardiac is going to be short cells that are connected to each other, kind of short boxy cells, versus your skeletal muscle, the muscle goes the whole length, or the cell goes the whole length of the muscle. Okay, does that make sense? Another way, functionally, you can tell the difference between a skeletal and a cardiac muscle is a skeletal muscle, you can remove all extracellular calcium, stimulate it with an electrode, and it'll still contract. And it'll contract for a while too. And that's because you don't need calcium to come into the cell for it to contract. And it'll have enough calcium inside the cell to contract for a while, but eventually that calcium will leak out and then you'll be back to not contracting. Cardiac muscle, on the other hand, if you take out extracellular calcium and stimulate it with an electrode, it can't contract. It needs that extracellular calcium. And that's actually how, I don't know if you've ever heard of Ringer's solution. Ringer's solution is a classic physiological solution you use for like most tissue to keep it alive. And the guy who came up with Ringer's solution was a guy named Ringer, not surprisingly. And what he found was when he used regular tap water to make up his solution, the hearts would contract fine. They're fine. And then one day he used distilled water instead, and his hearts wouldn't contract anymore. Like, what the hell's going on? What did I lack? Well, he had hard water, which had a lot of calcium in it. And when he made his Ringer solution out of that, the hearts would contract. But when he made it from distilled water, he had to add calcium back to get the hearts to contract. Ringer. Remember that. And remember who told you. Okay. I hate to tell you guys, but that's about it for today. It's only 1:45, but typically this is, I could go on and do smooth muscle contraction, but I don't have those notes written up for you yet. Should we go on and do smooth muscle? We've got one more lecture, which will be next Tuesday, and then our exam is Thursday. Any questions? I've got a game I'm putting together that I'll have up for you by hopefully by the end of the weekend. Something you can play to help you learn some of these vocabulary words. Okay.
Oh, I'm sorry. You said your exam was next Thursday?
I believe so.
On the syllabus, it was scheduled for the 23rd.
I have it for the 18th. I think I moved it up. No, wait, hang on.
Yeah, it's Thursday the 18th.
Yeah, I think I moved it up. It's Thursday the 18th.
Thank you.
Yeah, so Tuesday we will go over smooth muscle contraction, cell crawling, and amoeboid movement. And then we'll probably have an extra, half hour left over like we do today. We can do some review. Anyone want to do a review today on what we've already done? I don't know. Claire,
I would like to have a review, please,
right now, or do you want to wait till Tuesday?
right now.
Okay, good, good. Anyone who wants to stay for the review can. If you don't want to stay, if you're a know-it-all, you already know everything.
Would we be doing another one on Tuesday?
Yeah. I mean, these classes seem to be going kind of short compared to when I'm in class, so we'll have plenty of time. We can do a review on Tuesday as well.
Okay, and it's going to be reviewing everything or just today?
Whatever. I'm open to suggestion here.
Okay.
I'm here for you. I'm here for you. Whatever you ask me to do, I'll do within reason. Robin Russell, are you still here? Oh, Robin. Okay, do you have any questions for review? I mean, that's usually the way I do a review. You bring me some questions and I will do my best to explain them.
So, will we be able to find microtubules and microfilaments in smooth muscle cells, like in the smooth muscle, or is it just in the cells?
Yeah, microtubules and microfilaments are in all cells, and you've got certain ones that are called the non-muscle ones. I think I talked about non-muscle actins and stuff. They're in every single cell. They're involved in your cortex holding the plasma membrane together and other cytoskeleton things. Same thing with microtubules. They're in every single cell. But then when you get into skeletal and cardiac muscle, you've got some really specific actins and myosins that are there. But don't forget your skeletal muscles have all the other ones as well, because they're cells like any cell. What else can I tell you? I guess that's about it. Okay, anybody have any more questions? Preguntas?
You said the drug facts are going to be on the exam, right?
Yeah, I mean, sometimes I'll use the drugs in like a fill in the blank or something, and sometimes they'll be in an essay question, like what would happen to this cell at this state if you added this drug right now, and then you have to know the mechanism of the drug, and then make a guess as to what would happen to the cell if you use that. See what I'm saying?
Yeah, thank you.
So is the test going to be like multiple choice, short answer, and fill in the blank?
Yes, there's a test up there. There's a test up there you can look at, but typically about 50% of it will be like multiple choice, fill in the blank, drag and drop kind of stuff. And then the other half of it will be essay questions.
Is it going to be very similar to the structure of the test you have posted?
I believe so. Let me pull that up real quick. It's going to be a little different because it is online instead of in person. Where's my old test? Previous exams, exam 119. So I'm opening up. Why don't I just share it with you as long as we're here? It might be a half decent review as well. Sharon is Karen. Okay, so here's the one that's up in Moodle right now. And the first page is that essay thing or the assessment thing that we always do in biology. Okay, draw or describe the process of microtubule polymerization in vitro. Indicate the rate determining step. So we talked about in vitro versus in vivo polymerization. This would be a question like that. Now on our test, I wouldn't ask you to draw it because it's online and not everyone has drawing capability. So I'd probably just say, describe the process of microtubule polymerization. Make sure you check whether it's in vitro or in vivo, very different, right? And I could ask it either way. And then here are fill in the blanks. G protein responsible for filopodia, any proteins associated with cilia and flagella, blah, 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 blah. And then fill in the blanks of sarcomere structure. What the hell is that? I think there's supposed to be a figure here that isn't here. Yeah. So this is something I like to do a lot too, where you'll have a figure and different points on the figure pointed out, and you'll have to say what those proteins are. I don't know why it didn't show up here. I guess going from Microsoft Word to Moodle wasn't good. Name and describe microfilament structures that could get produced during cell crawling and tell the G protein responsible for it. Okay, so notice all those four were just kind of memorization of what we talked about in class. Very black and white, I said this, say it back to me. That's all it is.
Sorry, I just want to let you know, can you hear me okay?
Yeah.
So when you download the biology exam and then open it in Microsoft Word, the figures are there.
Oh, good.
Like when I open it on my computer, I see the figures with the
Excellent.
lines.
Thanks for telling me that.
Yeah. Can I ask you a quick question?
Sure.
So
Nope, only slow questions. No quick
yeah,
questions.
we gotta slow the class down. So, with the drugs, for example, like, the ones that prevent or block polymerization, is
Yeah.
that like what you would, like, why would you ever want to do that? Like if it was cancer cells?
Like I might say, you've got a cell that you've stimulated to crawl. We haven't talked about cell crawling yet, but we will. You've got a cell stimulated to crawl, and then you add this drug. What do you think is going to happen? And you will know that in cell crawling, you've got depolymerization and then repolymerization. If you stabilize the microtubules with one drug, it'll just stay still. If you hit a different drug, it may allow depolymerization and not allow polymerization, and you'll see the structure fall apart. So that's the kind of thing I would ask.
We need to know in the notes you provide and in the slides, or are there going to be things in the lectures and the textbooks that aren't there?
Everything on the test will be from either the notes or the PowerPoints. I usually don't take things from the textbook that aren't in class, but I may ask you in ways that are different from the way you saw them in class. See, this is a good example of that, I think. You're a cell biologist studying the kinetics of actin microfilament assembly in vitro. Okay, in vitro. You note that the rate of assembly can be accelerated tenfold by the addition of a cell extract. So if you're in vitro, that means you've got actin and ATP and you're watching them polymerize by themselves spontaneously. And now you're going to add something to that from a cell extract. Cell extract just means you burst the cell open, you take all the proteins and stuff and you add it and you see, yeah, does that help? So you add cell extract and the rate of assembly goes up tenfold. Something's helping it to assemble. This is despite the fact that you keep the levels of actin and ATP constant. So it's not because of ATP. What else is in there that's going to cause the assembly to go faster? And I would say profilin, right? Profilin, is it profilin? No, formin. Formin's the one that sits on the end and puts the things in. And then profilin is part of that structure too. So basically what I'm asking with this question is, in vitro, it's just ATP and actin. What else could you add that would make that go faster? That's something from in vivo. Okay, this cell extract is then grabbing things that are in the cell in vivo, and now we're comparing in vitro polymerization to in vitro polymerization. And this is kind of where a lot of these proteins were found originally. It's like they had this in vitro experiment where they're polymerizing actin, and then they'll take a cell, break it apart, separate all the proteins, add these proteins back, and see which ones are going to change the rate of assembly, and then they isolate that protein. And they're like, oh, look, this protein's important. That's the kind of experiments they used to do anyway. Okay, let's look at the next question. Imagine you are an embryologist studying an embryo from a newly discovered animal, a rodent of some kind. I say rodent because people don't mind killing mice, usually rats. Bad joke. You see a tissue that is clearly a striated muscle, but you can't tell through the microscope if it's skeletal or cardiac. Okay, this is Claire's question, because this is what you asked me. What's a simple functional test that you could perform to tell whether it's skeletal or cardiac?
Oh, I know this one. I know it. I'm going to try it.
Okay.
I got it. Okay. Add calcium to see if the muscles contract.
Right. Try and contract them without calcium, see if they contract. Add calcium back, see if they contract then, because that's your functional test between skeletal and cardiac. So you can see how I guess I did explain this in class, but it's just presented in a way that makes you think, what's going on here? And you basically, the answer is a hypothesis. Come up with a hypothesis that may, explain some of the phenomena we talked about. Here's another one. I'm not going to go over because I might use this on the test. What would be the effect of adding phalloidin to actively moving amoeba? Okay, so Carrie, this was the question I just answered for you. How would you use drugs? This is how I would use the drug in the question. Okay. And of course, since it's in Moodle, there may be just multiple choice or true false. I don't like true false, multiple choice or something. So you could see those as well. I don't have a Moodle test to share with you right now. So I won't be able to do that. Okay. Any other questions? Comments? Oh, Rachel's here. Still spelling your name wrong, Rachel, I see.
No, you're just pronouncing it wrong. Not pronouncing it wrong. You just don't accept that. That's how I spell my name.
How do you pronounce it Rachel?
Yes, I pronounce it Rachel because it's supposed to be Rachel, but my dad's name is Richard, so he decided to just spell it that way.
Oh, now it makes sense. Now it makes sense. Okay, I won't tease you about it anymore.
Thank you.
All right. Well, we'll see you guys on Tuesday. Have a great weekend and study cell biology. I'll send you an email when I got that game ready for you. Bye-bye.